not the presenter screen. Okay, so I've met most of you in person. For those of you I have not, I am Derek Belka. I am working with Dr. Andrew Schuler, and we are investigating microbial competition as a cause of process failures in enhanced biological phosphorus removal. So why are we looking at this issue? In the United States, the National Pollutant Discharge Elimination System regulates the discharge of phosphorus from water resource recovery facilities, or WRFs, which is where our municipal wastewater is treated. Shown in this graph on the right, we have this trend where over the last several decades, discharge of phosphorus from water resource recovery facilities is being increasingly regulated, while in the orange line, we are seeing a high level of violations even on a per permit basis. So there's obviously a disconnect between the professional practice of designing and implementing biological phosphorus removal and the actual outcomes that we see in the field. So what enhanced biological phosphorus removal does is remove phosphorus from wastewater. And it does that by selecting specifically for polyphosphate accumulating organisms, or PAOs, that take up a large amount of phosphorus. And to do this, their metabolism relies on cycling them through anaerobic and aerobic reactors. And this cartoon above is an example of a process that could be used to accomplish enhanced biological phosphorus removal. And this reduces the effluent phosphorus from these facilities down to a level usually less than around 1 milligram per liter is pretty common. So why do these facilities fail? One of the current thoughts out there, ideas out there, is that competition for limited organic carbon, specifically volatile fatty acids, between PAOs and another volatile fatty acid utilizing organism called glycogen accumulating organisms. And they compete in anaerobic reactors, and this leads to EBPR breakdown. And the reason that people have come to this conclusion is because we've conducted primarily lab experiments in the past, like this one on the left, where people have shown that successful EBPR is basically PAOs outcompeting GAOs for volatile fatty acids. And so in doing so, the GAO population is suppressed by the success of the PAO population. But a lot of these researchers have also shown that changes in reactor conditions, so pH, temperature, substrate availability, a bunch of these different things, can cause the inverse of this, or failed EBPR, where we have the GAOs now outcompeting PAOs for volatile fatty acids and suppressing the PAO population. So this is sort of the general idea of why these systems fail. And so it would stand to reason that if we go to the full scale system, in a successful EBPR plant, we'd have an abundance of PAOs and essentially no GAOs, and we'd have the inverse in a failing system. But when we look at data from full scale systems, we actually see that the presence of GAOs does not seem to affect enhanced biological phosphorus removal performance. And actually what we see is that an abundance of organic carbon, which is here represented as a COD, or chemical oxygen demand to P ratio, greater than about 60, seems to be more correlated to a rise in the abundance of GAOs within the system. And so we're seeing, essentially a successful system with GAOs present in it. And this suggests a number of other hypotheses other than this sort of competition model for failure. One of those is that the GAOs are just opportunistic. So they're only coming into the system when there's an abundance of organic carbon available. It also suggests, well, maybe the competition interaction is actually occurring and there's other PAOs in the system that are important contributors. So they're using something other than volatile fatty acids to accomplish phosphorus removal. It also, that leads into the idea that, well, these interactions are probably more complex than this simplistic back and forth. And it also suggests that maybe a less selective system could be more resilient, because we don't want to have our main group of functional organisms being completely suppressed by a competitor at any given time. So how are we looking at this? We're currently operating 2 reactors in the laboratory, and we're going to operating 2 reactors in the laboratory to select for 2 different groups of PAOs. One group uses volatile fatty acids, the other group uses amino acids, and we're going to start subjecting these reactors to various different failure conditions. And that will hopefully help us elucidate, okay, how are these interactions actually taking place in EBPR reactors, and what kinds of other interactions could we be seeing that could be contributing to a successful EBPR system. There's some really great references out here on this. It's a pretty widely studied topic, so there's some really good papers here to go read. And I want to say, give a shout out to Andy Schuler, my advisor, Kaylin Fisher, who's been a huge help navigating the lab during COVID, and my undergraduate assistant, Haley Ormsby. And now I'd like to open the floor for any questions if anyone has them.
Good work, Derek. 2 questions. One, I was wondering if, have you looked at the effect of microbial abundance on phosphorus remover in your systems? And 2, how are you planning on scaling your miniature scale reactor in the lab to, like, a larger scale reactor in like a wastewater treatment plant?
Sure, those are great questions. So to address your first question, the abundance of, so it's kind of, one of the things that's hard to tell is everything's a relative abundance. So it's number of PAOs divided by number of total bacteria, or number of GAOs divided by number of total bacteria. And so we don't, the relative abundance, we don't really see like an absolute change in the amount of phosphorus removed as the PAOs go up or down. So if you remember back to that that graph I was showing, the quantity, or the number of PAOs in that system were kind of moving around. And we can't really say that we had, exactly 20,000 PAOs in that system. We just know that we had 5%. Does that make sense? And so then to address your second question, one of the things that we can do is, even though we're operating batch reactors in the laboratory, we can manipulate the way we feed and waste from them to act more like a full-scale system. So one of the experiments that one of Dr. Schuler's previous graduate students did was to, rather than put all the carbon in all at one time and sort of a big slug at the beginning of our anaerobic phase, which would be the first thing that the first basin encountered by the influent coming into the process, we feed it in really slowly so that we keep the concentration more like it would be in like a large continuously mixed flow reactor as opposed to a batch reactor. So it kind of gives at least some simulation of what the real world conditions are like. And then we can move that up to pilot scale, and then maybe even up to a larger system than that.
Great work.
I have a quick question, Derek.
Yeah, I think we can do one more, and then we got to move on to
All right. Yeah, actually, I have like a dozen questions, but I'll just corner you one day in the lab and get through.
Sure.
I think the one most like pressing for me, you showed there's an anaerobic, anoxic, and then aerobic with the recycle. Is that typical for this? Is this also used for nitrification, denitrification? I mean, it just seems. Is it an anaerobic process, or why that particular setup?
Yeah, so you need both. Like, you need an anaerobic period, and then you need a period where you have an electron acceptor. So the process that I showed, it's very common to see phosphorus removal and nitrogen removal coupled together in a treatment process. Usually the first thing that happens, BOD removal is required in basically every single treatment plant. Then people start regulating nitrogen, and then phosphorus usually gets tacked on after that. And so it's just like you keep adding on to the process. So the anoxic basin is not, it can participate in phosphorus removal, but the graphic that I showed is just an example of one kind of process that does nitrogen removal and phosphorus removal.
Super, thank you.